All right, welcome to today's seminar. Our speaker today is Dr. Anand Astragiri, who is assistant professor of in the Division of Chemistry and Chemical Engineering at Caltech. Dr. Astragiri got his bachelor's degree in chemical engineering at Cornell. Then he went to MIT to do his PhD in chemical engineering as well, with a minor in molecular cell biology. After that, he postdoc at Harvard Medical School in cell biology for a couple of years before joining Caltech. Dr. Astragiri has been is currently an associate editor in the PLOS, Public Library of Science, computational biology, and he was a recipient of the Concern Foundation Award for Cancer Research, and he chaired the Engineering Cell Biology Conference last year. He works in the area of cell molecular bioengineering, and he's going to talk to us about how cells organize themselves to form multicellular structures, and that's his main research interest. So, Dr. Astragiri.
Thank you. I'd like to thank Michael for the invitation and the department for hosting me. I've enjoyed the conversations this morning. I'm looking forward to the rest of the visit. What I'd like to share with you over the next maybe 45, 50 minutes is some of the work we're doing at Caltech on what I call integrative morphodynamics, long-term, really, to just describe our interest in multicellular structures, how they form, and how we might be able to engineer these multicellular structures. Part of the reason, of course, we're interested is just the aesthetic nature of the problem. I think nature presents a number of remarkable structures, some of which you can now find on the web. And it's remarkable how cells are innately capable of performing the processes needed to form such structures. But in addition to that, we have a number of practical interests that drive us, and one of them is related to cancer development. Typically, when one thinks of cancer development and disease progression, it's fairly clear now that there are molecular and epigenetic perturbations that drive the disease, the progress of the disease. But in addition, what is also known and very clear clinically is that the tissue of interest undergoes a number of multicellular and morphological changes. In fact, it's these morphological changes that are often used for early or late detection and for diagnosing the potential course of the disease in future time. And so what I'm showing you here is a typical mammary tissue where these soccer ball-like structures have epithelial cells at the defining the shape of the structure, and the milk proteins are secreted into the lumen. So there's an apical to basal polarity of these cells. And that structure helps provide function and encode and maintain function. As the disease progresses, the structure goes awry, not in a single way, but in multiple ways. And what one would like to know is what are the molecular changes that correlate to the different types of structural perturbations you might see. This is useful from the context of diagnosing what might be the underlying reason you see a certain structure. But if you also understand what molecules give rise to structures, you might be able to shunt structures towards states that have better prognosis than others. So there's a therapeutic reason to be interested in multicellular structures. And we've also been thinking about not only how structures go wrong, but how structures can be made to go right, and that is in the context of engineering tissues themselves. I'm showing you just one example here where what's being depicted is the organization or the self-assembly, if you will, of individual endothelial cells on the top to a proper vascular structure that could guide blood vessel flow. This is obviously a multi-step process. It involves the initial aggregation of cells, the growth of cells in certain locations and not in others, so that you get a hollow tube and not a filled-in tube. And obviously that structure is important for the downstream function. So the question in our lab has been, how is this multi-step process of forming a tissue guided by the program that cells execute? Okay. Now, if we want to execute these kinds of steps and produce tissues in engineering examples, one needs a scaffold around which cells would organize, and there's tremendous advances currently in the area of biocompatible materials that would house the engineering of tissues. Cells would interact with these materials, synthetic materials drawn here as these white fibrous material that can be derivatized with specific signals that cells recognize. Their mechanical and physical properties can be tuned for the local situation, the local tissue of interest. And oftentimes these parameters can be changed dynamically. So we have a remarkable level of control over how these polymeric scaffolds can be designed. But the question of interest, or a challenge in the field, is how would cells respond to those scaffolds? How would they, in combination with their intrinsic rules for assembly, work in the context of this synthetic scaffold to form a tissue that you might be interested in? And so our lab has been interested in two broad sets of questions. One is the identification of the principal molecular signals and the rules with which cells use these signals to form multicellular structures. What are these signals and what are these rules? And if we think we know some of these signals and rules, there will surely be more than one or two. It's going to be a complex set of rules and molecules. One needs to think about an integrative framework with which these rules prescribe a certain structure, because ultimately you want to go in and change a particular molecular pathway and alter the nature of the structure that you form. Okay. So the first question alludes to the kind of experimental work that goes on in our lab, and the second, of course, is pointing you towards a computational challenge of integrating this information to make it predictive and to guide design. And I'm going to touch on both of these questions, beginning with the first, and focus on one system that we use in the lab, which are these human epithelial cell systems, because of its relationship and importance to cancer. Majority of human cancers occur in the epithelium, and one of the normal constraints on what cells do, epithelial cells do, is this idea of contact inhibition of proliferation. It's generally said that cells, epithelial cells in contact with one another, will be inhibited in their division, and when that constraint is relaxed during the progression of disease, you get an accumulation of cell mass and the formation of a tumor that can then further evolve into metastasis and a full-fledged lethal situation. So we've been interested in this issue of contact inhibition of proliferation, and this phenomena has been studied by many others. We know, for example, that certain molecules at cell-cell interfaces called cadherins, in this case epithelial or E-cadherin, is involved in inhibiting division of cells. Among others, Chris Chen at University of Pennsylvania has studied this phenomena using micropattern surfaces where these epithelial cells are put on an adhesive square surrounded by a non-adhesive region. And what he showed in that particular paper is that in a cadherin-dependent manner, cells on the periphery will undergo cell cycle activity. They light up with this particular stain that looks for the synthesis of new DNA. But the cells on the interior do not. So cells on the interior that are surrounded by neighbors are contact inhibited, while those on the periphery display this cell cycle activity. Okay. The question we've been interested in is if you take any normal endogenous situation, what you typically have isn't just cell-cell contact, but also other molecular signals in the environment. An important class of them are soluble growth factors that will tell the cell to do exactly the opposite, which is to divide. So the question is, how do cells reconcile what seem to be opposing inputs, one to divide and one to inhibit? Is this moniker, contact inhibition of proliferation, sufficient to describe this balancing? Okay? Or is it more quantitative, that they're weighing the level of growth factor against levels of contact? And if it's quantitative or if it's not, if it's just inhibition of because they're in contact, what are the pathways that are involved, and can we begin to tune them towards engineering objectives? So we started this work by doing quantitative experiments where we looked at a two-dimensional monolayer of epithelial cells. These cells organize into clusters, okay? And you can visualize those clusters by staining the nuclear material of these cells using DAPI. And we also show here, stained in green, is this DNA synthesis marker, BRDU. And we do this in cells that are treated with a soluble growth factor, EGF, that promotes the division of these cells. Okay. And what we noticed in these non-transformed, non-cancerous cell line is that at early time, even the cells in the interior undergo DNA synthesis and are active in cell cycle. Okay? So these cells that should be contact inhibited because they're not transformed exhibit a uniform mode of division in this early time. Only later in time, the peripheral cells are the only ones dividing, and the interior group of cells are contact inhibited. Okay? So there's a temporal aspect to when contact inhibition kicks in, even in the normal cell systems. And you can quantify this as shown in the bottom, where we're looking at the percentage of cells that are on the periphery or in the interior that are undertaking DNA synthesis. And only at the later time point, at 72 hours, you see a spatial difference, okay, a pattern. Now, if you change the experiment so that you start with a lower dose of EGF, so the top bar graph is what I had in the previous slide. Now, in the bottom bar graph, I'm introducing EGF at 0.1 nanogram per mil. Okay. The onset of the spatial pattern occurs earlier. It occurs at 48 hours instead of 72. So there's not only a temporal element, but it's tied into how much of EGF is presented to the cells. And all of this is indeed contact inhibition. That is, the spatial pattern that you see at 72 or 48, okay, is driven by contact inhibition, because if one goes in and blocks the coherent interaction between cells using an antibody, you eliminate that spatial pattern. So what looks like a pattern here at 0.1 nanogram per mil would become equal-sized bars when treated with ecoherent antibody. Okay. So the question naturally is, why this EGF dose dependence and what's happening as a function of time to make contact inhibition appear? Okay. And one of the things that had been known about growth factors like EGF is that they bind to receptors on the surface of the cell, and then those receptors are brought into the cell and the growth factor is degraded through a process called trafficking and endocytosis. So we would not expect EGF to be always at 1 nanogram per mil or 0.1 nanogram per mil, but rather decay over time. So we try to measure the amount of EGF in the medium as a function of time, and indeed it decreases over time. What's important to notice, though, is that if you look at the time points when the two cultures exhibit the pattern in proliferation, those two time points occur at roughly similar levels of EGF. So the cultures, no matter how you start them, get past this common level of EGF, at which point they exhibit the spatial pattern. Okay. And this has been verified in another approach, a time-invariant experiment, where you start initially the culture not just at 0.1, 1, or 10 nanogram per mil, but over an even broader range. And you can confirm that it's only when you get below this concentration of EGF that the spatial pattern is exhibited. Yes?
So I imagine the concentration of EGF would be different in the center of a cluster of cells versus in the periphery, and perhaps relating to why you see this difference. When you measure this way, are you measuring an average level of EGF, or can you tell what the level is in the medium?
Yeah, that's a good question. So there's a potential for a spatial gradient in EGF is the question. This measurement is a global measure in the medium. And I want to address that spatial pattern issue in a couple of ways. One, keep in mind that this is a two-dimensional cluster of cells. So they're sitting on a dish over which the medium sits. So all the cells are being exposed to the medium. Okay? Nevertheless, if the binding rate is high enough, you can get a transport-limited spatial gradient in EGF that sits above this two-dimensional cluster. But we don't think such an event is occurring. We haven't measured it directly, but we don't think that's occurring because, as I showed in the previous slide, the spatial pattern and division we see is very discrete to the outer rim. So it's not a gradual phenomena that seems to occur. And furthermore, the E-cadherin inhibition, that's the protein mediating cell-cell interactions, eliminates the spatial pattern. So even if there is a spatial gradient in EGF, the contact inhibition is critical to generate that pattern. Yes. That's a possibility, that there's a sort of a spatial constraining of the interior cells, as you said, where the top layer that's exposed to the environment is smaller in the interior than it is in the periphery. That's a possibility. But even if that were true, you would still need coherence and contact to enable that phenomena. So what we're implicating is not that coherence are the direct regulator of proliferation, right? But they must be there upstream to enable contact inhibition. But that could be occurring by physical constraining of the cells. Yeah. Even with a uniform level of EGF. Yeah. Right. So according to this model, so let's go with that possibility, that there's an exposure issue. But that issue will kick in only at this threshold. So only when the bulk level of EGF dips below this threshold, the cells in the interior undergo this inhibition of division. And we reasoned that that might be because the inhibitory effect of coherence and contact is at a certain level, and only when the growth-promoting effect of EGF goes below a threshold, the inhibition effect can overtake it. Okay? Possibly by shape changes. And so it's sort of this threshold model that the EGF level has to dip below a threshold to go from this mode to this mode of division in this non-transformed cell system. And so we would suggest that this might be a quantitative titration of the level of growth factor against a certain amount of inhibition imposed by cell-cell contact. And to cut through a couple of different experiments, we looked at pathways that might be mediating this phenomena. So we know that growth factor triggers various intracellular pathways, MAP kinase, AKT, that controls cell cycle. And so one can ask, at this point in the situation, when you have contact inhibition, which of these pathways, or perhaps all of these pathways, become impeded and are not able to function properly. And so when we look at MAP kinase, we see a loss of signaling in the interior of these clusters, whereas in AKT it's more uniform staining. So it suggested to us that contact inhibition is working specifically through the MAP kinase pathway and inhibiting this pathway while not affecting the AKT pathway. Now, everything I've said so far suggests that growth factor is dropping, and when it gets below this threshold, all this stuff kicks in. Contact inhibition, loss of our signal, and so forth. If that's true, I should be able to dial EGF back up and revert everything, which is what we observe. So if you add EGF back into the culture, you can kick this system back into this mode of division. Okay. So in this model, we're suggesting that there's this quantitative balancing between contact and growth factor. The implication of that is, if it's truly a balancing act, if one increases the level of cell-cell interaction, you should be able to change the threshold. So if you make the inhibitory effect even greater, okay, then the inhibition should be able to overcome EGF when EGF is even higher. Right? And so we try to test that idea by going back to the type of micropatterns that Chris Chen's group had worked on. So these are 200 micron adhesive patches into which, these are all, again, two-dimensional patterns. We coat either a low number or a high number of cells. And this is a very simplistic idea that if you put more cells into the same area, they will have to increase the level of cell-cell interactions in some physical way. But cells are not going to be PowerPoint bodies, okay? So that sketch is not accurate. They're going to be ruffling, their membranes are going to be curving around each other. Some of them could be sliding underneath a neighboring cell, okay? So there's going to be a variety of different issues going on, but overall, the level of cell-cell interactions will be higher here than they are here. We can verify that there are more cells here by looking at that nuclear stain DAPI. And what's important is shown here. If we treat the cells with a very high dose of EGF, larger than anything I've discussed so far, 20 nanograms per mil, we can induce a spatial pattern in division. This is the BRDU DNA synthesis marker in this high density of cells. Okay, so enhancing cell-cell interaction induces the spatial pattern at a much higher level of EGF, suggesting the threshold has increased. Okay. And this too, given all the artificial nature of this experiment, that it's on a micropattern and all the nuances I described earlier, if you knock out cadherin using an siRNA, you can eliminate this pattern. Okay, it goes back into a homogeneous proliferation. So this, again, is contact-mediated inhibition of proliferation, occurring at a much higher dose of EGF than previously shown in the slides. So this led us to a simple sort of state diagram representation for how cells are of the two states that the cells can exist in, either a contact-independent mode of division where all cells in the cluster have a uniform chance of dividing, or a contact-inhibited mode where the division is restricted to the periphery of the cluster. So if you pick this dot here and call that your system, what I'm suggesting is that as the growth factor level dips below a threshold, it will shift into this contact-inhibited mode when contact inhibition overtakes the growth-promoting effect of the factor. If you enhance cell-cell interaction, it will again undergo this transition, but the transition won't occur at the same level of threshold anymore. It will occur at a higher dose. So the transition line actually has some non-zero slope of that type. Okay. So if this is all true, okay, it has certain implications for growing cells not on micropatterns, but just on a dish, as I was showing you earlier. And the implication is that if EGF level, now on regular dish of cells, if the EGF level is dropping over time, as I showed you before, if I increase the level of cell-cell interaction, then the EGF threshold should go up, according to that state diagram, and the transition should occur at an earlier time. Okay. So we tested this idea by going back to cells stimulated with that 0.1 nanogram per mil of EGF, when normally you get the spatial pattern at 48 hours. Now we're looking at them at 24 hours. In control cells, there is no spatial pattern. But if you overexpress E-cadherin, the molecule that's mediating cell-cell interaction, you can now induce spatial pattern at a much earlier time, 24 hours instead of 48. Okay? So it's consistent with this state diagram model for contact inhibition. So we think this idea of a state diagram and this ability to switch between contact independent and contact inhibition has implications in various areas. One of them is in the area of development, where one of the interesting questions has been how a size of an organ is determined. So in development, you often have a field of cells or a growing field of cells that is induced or is exposed to a spatial gradient in a factor, typically called a morphogen, or in this case a growth factor. Some morphogens can induce the division of cells. It's quite clear that this gradient will eventually taper off to some very low level, and therefore the cells out here won't grow anymore. But the question is, what stops the cells over here that are close to the source from growing? And one argument provided by Boris Schreimann at UC San Diego was that he argued that in a theoretical model that perhaps there's a buildup of what he called a local pressure term, that as the cells divide, there's an accumulation of cells and a local pressure that builds up that desensitizes these cells to the growth factor, so they don't divide anymore. We would suggest that some of the experimental results we've shown here is exhibits that type of desensitization, that as you enhance cell-cell interaction between neighboring cells, the cells will actually desensitize to the soluble growth factor by increasing that threshold growth factor level. In the context of cancer, which originally motivated this work, the way we've been thinking about this is that if we think of normal non-transformed cells as being contact inhibited, then in order to transform, this cell has to get over this threshold in the growth factor axis to become contact independent. But what this state diagram would suggest is that you could have latent transformation, where cells are quantitatively moving to the left and getting closer to transition point, okay, so that the bump that they would need is smaller. Okay? So this is a way in which contact could be synergistically working with growth factor so that you slide down this axis so you can jump up to transformation more easily along EGF amplification, for example. So this was an interesting concept, and more recently, in unpublished work, we've tested and we think we've shown this type of quantitative transformation that is phenotypically, however, latent, because the cells always sit in this regime. And the way we went about testing this is to look at one of the analog perturbations that occur during cancer, and which is sort of most generally experienced just by tactile sensing. And that is that the microenvironment of a developing cancer becomes stiffer. We can emulate that stiffening in our experimental context now by plating cells not on a plastic surface, but on a polyacrylamide gel in which we vary the cross-linking density. Higher cross-linking density makes the material stiffer, and lower makes it softer. This is a technique developed by Yuli Wang now at Carnegie Mellon and used broadly to study the effect of stiffness of microenvironment. When you put these cells on a soft surface, elastic modulus of 7 kilopascal, what we notice is as you dial up EGF level, you go from a contact inhibited to a contact independent mode of division somewhere between this jump, 1 to 10 nanogram per mil. Because here you have proliferation restricted to the outside, here you have uniform division. When you stiffen the matrix, this transition occurs at a lower dose of EGF. So between 0.01 and 1, you now shift the system to this uniform mode of division. So this is sort of consistent with the idea that the system was originally sitting here on the soft, but then the threshold has dropped as it has slid closer to that transition line. So we would suggest that microenvironment stiffening quantitatively modulates contact inhibition, and you can follow that modulation not by looking at it phenotypically, but by measuring this threshold that's being dialed down. Now, there are other implications of this diagram that I won't be able to get into, but what is also suggested here is that contact and the nature of contact is changing. And we have evidence for that by doing confocal microscopy of these cells and looking at where E-cadherin and another molecule I haven't talked about, ZO1, are localized, and we can see changes in the maturation of that contact as we stiffen the microenvironment. So the system is moving to the left on this axis. So what I've described so far is what we think also just one building block in many that are needed for the engineering of tissues. So the example that I've been emphasizing is this proliferation that in principle now can be switched between at a rate that grows proportional to the area or proportional to the perimeter. And our most recent results suggest that even mechanical compliance of the substratum can affect this mode, this switching. Okay? So we think this could be a useful way to regulate colonization of a material, for example, if you're interested in growing clusters on synthetic surfaces, and also to localize who divides and who doesn't. But building tissues is going to require more than just growth of a cluster. One would like to think about how do you make a cluster in the first place. And so this notion of aggregation has been of large and wide interest in, especially in developmental biology, for example. And one of the ideas that's out there is this notion of differential adhesion, where if we look at cells interacting with each other and with the surface underneath, the argument has been that if the cell material adhesivity is greater than cell-cell adhesivity, then you would reduce the tendency to aggregate. If cell-cell interactions are more favored, that would promote aggregation. So if you look at aggregation and as a function of the adhesivity of a material, which is something that an engineer, a polymer synthesis synthetic chemist can dial in and out of a polymer, okay, by just changing the number of green triangles or ligand on the surface, what one would expect is as you increase the adhesivity, you should decrease the aggregation. But this is all based on sort of an equilibrium argument. Okay? But if you look at videos of cells aggregating, it's obviously dynamic. This video is over about a 16-hour period. Initial cells isolated, collapse together on a two-dimensional surface to form two-dimensional clusters. This involves both a transport step of finding one another and, once found, hanging out together long enough so that a third member can join. So there's a time scale of transport and a time scale of local reactivity so that the pair that has formed doesn't fall apart too soon relative to the motility time scale required to bring a third party in. So Melissa Pope in the lab has been measuring using fluorescent imaging. This is a phase contrast video, but using fluorescent markers of the nucleus and the membrane to measure aggregation dynamics. And what she's observed is that there are situations where transport can limit aggregation dynamics, and you can actually get a biphasic trend as a function of the adhesivity of the surface. This biphasic trend is based on the fact that motility of cells is optimized at an intermediate level of stickiness to the surface. Okay. So we would suggest that aggregation may not behave monotonically, but also biphasically, and whether you're in the monotonic regime or the biphasic is discernible, depending on which step is limiting the aggregation dynamics. And more recently, we've been thinking about ways in which we can bring cells, isolated cells, to a common area so that they can undergo aggregation and then grow. So just as another building block towards, of many that are needed for tissue engineering. And we've been conducting this work in the context of micropatterns, where it's motivated broadly by the fact that a migrating mammalian cell is typically described in this with this shape, where it has a broad leading edge that reaches out and grabs onto the surface, generates contractile forces within the body, and lets go in the rear, which is the narrow tail that's shown there. Several groups have been motivated by this general morphology to suggest that perhaps if we emulate that morphology on a micropattern, we can force cells to have a direction. Okay? On a uniform surface, cells will move for a while in one direction, then switch and move in another way, exhibiting a random walk. So the idea has been to use micropatterns to force cells to know or force them into a situation where front is defined and rear is defined. And the shape that's been popular is this teardrop type of shape. And one of the things we've observed is if you arrange these teardrops in the fashion shown here, and you put the same cells that I've been talking about so far on them, these are mammary non-transformed epithelial cells, they will run around on the pattern. I think this one will go about 8 times in a highly persistent manner. Okay? So 80-20 is the statistics over a large number of cells. But and what we learn from this, okay, is that these cells tend to jump out of the tip and not out of the blunt, and they jump to the side, right, onto a blunt end. But if you change the nature of the pattern so that you make the tip, in order to jump out of the tip now, the cell has to go head on into a blunt. Okay? The only sideway jump that's permitted is to go from the blunt to the tip. The cells are ambiguous. So they will move far slower, they're conflicted, and this one will change direction after maybe 2 jumps. The overall time of the video is the same. This is about a 10-hour video, each of them. And the statistics here is about 50-50, 60-40. So from these types of analyses, we think the cells in this particular context prefer to jump out of the tip and make a sideways jump onto the blunt. Okay. But we're not comparing apples to apples, because this tip lands onto a blunt, but a sideways jump from a blunt to a tip is not shown on this pattern. A sideways jump is only permitted from a blunt to a tip here. Okay. So if we designed a pattern that would allow head-on comparison, no pun intended, between those two situations, and what we find is indeed the preference is to jump out of the tip and onto the blunt. So this is a yin-yang pattern where only sideways jumps are possible, either from a tip to a blunt or the other way around, blunt to a tip. And the preference is highly skewed to a tip to a blunt jump, 90-10. What's really interesting about this is that the movement patterns are tunable. Okay, so if you take this cell that was ambiguous on this pattern, but highly oriented here, and if you down modulate a signaling pathway known to be involved in motility, RAC1, you cannot knock it out. You have to dial it down to about 50% level of expression. If you knock it out, this activity will be gone. The lamellar extensions will be out, and migration will just stop. But if you dial it down moderately, you can now induce highly oriented movement where cells were not so oriented before. And this oriented movement goes away. Okay? So we think that these are really interesting ways to direct cell movement by manipulating pattern and refine movement, or manipulating intracellular signal to alter the nature of movement. We are, in addition to picking the direction of movement, you'd like to control the flow rate of movement. So if you take these sort of teardrops and you change two aspects about them, either the offset, which is how this pattern sits relative to this one, so I'm sliding this to the right here, or slide it north, okay, make the gap distance greater, you change the decision making at this pivot point. This allows more of a graded change in the flow rates, whereas this is very switch-like. It's even distribution, and then you just lose the connection to the north as the north spacing increases. Okay? So there are sort of fine control and strong control of splitter designs for controlling flow rate of cells on these kinds of surfaces. So assembling these parts could potentially give us access to sort of moving cells around with different flow rates and directionality. And as a way to think about that, we designed an attempt to move cells from one compartment to another. These compartments in this control surface are joined by a long rectangle, high aspect ratio rectangle. This is a millimeter, okay? Or connected by those teardrops that I showed before, but now opened out. Okay? So the cells should run from the bottom to the top. And the video here, well, the bar graph tells you that that's what happens. If you have on the control, there's not a segregation, but on the red bars, you get about a twofold enrichment of cells towards the top. And the video just shows you that movement. The red dot here correlates with the red dot there. We're just looking at the top half of this situation. The cell runs up, bounces back down, and then it will eventually come back. And some cells will escape down, but they will go back up. Okay, so they join onto the top reservoir. Now what you notice is some cells do escape, and they get considerably far down. You notice two cells interacting here and shifting position. So the point is, this is by no means a precise programming of every cell to go marching up to the top reservoir. You're going to have cell-cell interactions that you don't expect. You're going to have leakage out of the reservoir you're intending to collect in. But on average, you're generating a scenario for cells to move up. Okay, so these types of design ideas, some of them sort of molecular based because we have some understanding of a pathway. Others are more physical, either micropattern or perhaps the mechanical compliance of a matrix. But what we're trying to derive is how cells interpret these molecular and physical signals in a quantitative way to decide a state of performance, division, movement, and therefore affect multicellular outcomes, aggregation or growth of a cluster. And we think that these sorts of building blocks individually are the types of things one needs to bring a rational approach to tissue engineering. Now, what these building blocks also open up is the possibility of developing predictive models. So I'd like to spend the last, hopefully fast 10 minutes trying to highlight this modeling aspect. And what it opens up is the possibility that you have cells performing multicellular organization, each cell deciding a state based on a state diagram that you quantitatively understand, who are then, the state of which is then dependent on molecular cues. And this type of a description could give rise to a mechanistic lattice model that predicts the outcome of a multicellular system, and could open up questions or answering questions of this type. What are all the structures that are possible? How does the system explore phenotypic space as you play with certain parameters? And what are all the mutant structures that are accessible to a system? And I think these are the kinds of questions we want to answer in the context of cancer. We've been looking at it in the context of C. elegans initially because of the following advantage, and that is, in C. elegans, we understand fairly clearly how the adult organism comes about from a single fertilized egg. The adult organism, 959 cells, are the endpoints of this tree. And for certain snippets in development, we also understand the network that presumably gives rise to that pattern. Okay. So we can undertake the challenge of simulating or encoding in some mathematical or computational framework the mechanisms and asking whether it predicts the outcome we think it encodes, and what else is possible from those mechanisms. So we focused on a system where 6 precursor cells in the C. elegans system differentiate to give rise to the bulba. So the center cells divide to form 8 daughters, called the primary fate commitment. These 2 cells produce 7 daughters, called the secondary commitment. And these 3 in the outer ends commit to a tertiary fate and divide only once and fuse to the synctium around the tissue. So these cells actually form the orifice. These cells line up with the muscles. These fuse to the surrounding tissue and don't contribute to a vulva. So this is important to get a single opening vulva. If you have mutations in the system, you can get variants of this type, variants from this wild type. Okay, there are 2 factors, extracellular factors, known to drive this differentiation. One is an EGF-like factor that sits as a spatial gradient over these cells. And another is a cell-cell interaction, a lateral interaction between notch and delta. Okay? So here again, you have an interplay between EGF soluble factors and cell-cell interaction, giving rise to now this spatial pattern of 3-3-2-1-2-3, which is the wild-type pattern. If there are mutations in the system, like you overexpressing Lin12, which is the cell-cell receptor, you can get all secondaries. If you knock out Lin3, the EGF-like molecule coming from the anchor cell, you get all tertiaries. Okay? So we have a good understanding of various mutants that can form in the system when you perturb the molecules involved. What's important is, though, that these pathways, these 2 molecules, are not acting independent of one another. Okay? They are coupled through this reaction network that I've described here. EGF activates a pathway that controls notch and delta. Notch and delta activates pathways that control EGF signaling. So it's a bidirectional communication between these receptors, which we sought to model using a coarse-grained model where we track the activation of a pathway, MAP kinase, that is responding to the EGF signal, and the level of notch activity, the lateral signal that's communicated by the cell-cell interaction. Both of these are coupled, as I mentioned before, so you can see that there are certain terms in the differential equation that involve both factors, M and L. Now, that aspect of the model was something we analyzed just to look at the biochemistry of the signaling pathway. It predicts concentrations of signals in individual cells in arrays of 6. But that's not the prediction that we can actually go and test, because we cannot measure the activity of MAP kinase or Notch within individual cells directly. What we can see is the phenotype cells acquire, 332123, a normal vulva, or something else, and an abnormal vulva. So we wanted to map this onto the fates that cells acquire. And we did that with the following mapping scheme, where we said that the level of MAP kinase that's activated by EGF and the level of Notch activity determines the fate choice a cell will make. The absence of both sends them to a tertiary default state. If you have high EGF and MAP kinase signaling, you can get primary. If you have high Notch signaling, you get secondary. And if you have both of them, you have a mixed outcome. Now, these assignments are not arbitrary, right? So these are based on genetics that I showed you earlier. If you overexpress Notch activity, you get all secondaries. If you overexpress MAP kinase high enough, you will get all primaries and a multi-vulval system that has many orifices. So it's the most conservative interpretation of genetic data. And with this, we have a way to now predict phenotype at every parameter combination in the system. There are 8 parameters in the system. We pick the values of each parameter. We can compute whether that dot is going to be 332123 or something else. And we do that for every combination of parameter points in this 8-dimensional space. And we now have an assignment for what phenotype occurs at these combinations of parameter values. So somewhere in that space will be wild type, and in other places will be different mutants. And this has allowed us to answer questions that are sort of fundamental. If you have a given developmental network, how many phenotypes can it make? Is it so robust that it makes only one, or maybe a handful of others, or can it make much more? Can you get a lot of phenotypes by just quantitatively varying how a network performs? Okay. Are there novel phenotypes that people haven't seen in experiment? And the most mundane question is, hopefully, you're predicting experimentally observed mutant phenotypes. Okay? And I'm going to actually focus on the last one, because that in itself opened up a can of worms that had some interesting conclusions. So are experimentally observed mutants predicted? And importantly, are they predicted when you dialed the EGF axis up or down? Okay? And so we looked at that, and we said, EGF level can be increased or decreased, and these are all the phenotypes that are predicted by the model. In blue boxes are the ones that are observed in C. elegans. So the model vastly overpredicts the things that are observed in C. elegans. And so this raises questions about why you get all of these extra predictions of phenotypes. Okay? And one of the possibilities we were looking at is that C. elegans has related species that also make the vulva, and they make it by defining a 332123 phenotype for those precursor cells. Even more, they use the same parts. EGF is involved, MAP kinase is involved, Notch delta is involved. And an interesting experiment, a single author paper by Marianne Felix, and what she did there was she took these 11 species that all have a common wild type, and she either cranked up or knocked down the EGF signal. And what she observed is that even though they have a common phenotype as a wild type, when you dial it up or down, when you dial up or down EGF, now you start to see differences in differences in phenotype. Okay. Of course, if you go to the extreme, they collapse back to common phenotypes. So the yellow arrow here is one species, blue is another, and red is another species. I don't have all 11 here. So one hypothesis of these results was that, well, perhaps these species, okay, C. elegans, C. briggsii, C. remini, they're all wild type, okay, but they're quantitatively different from one another. So not only do they share the same parts, but they've actually wired them up in the same topology of a network, okay? But those networks operate quantitatively different. So that C. briggsii occupies this region in parameter space, C. elegans some other region, And because they occupy different regions, they have access to unique phenotypes when you perturb them away from wild type. Okay? It's an interesting hypothesis, but almost impossible to prove by experiment, because you'd have to go into all 11 species and show that this network and this network alone operates in each one, that there aren't additional arrows that might be there. And you have to prove systematically that this network is sufficient for generating the phenotypic diversity that you see. And the sufficiency criterion is very hard to prove experimentally. But in a model, that's exactly what it tests. So we went back to the model and we said, well, this model was designed with only that network. That was quantitatively varied. What are all the phenotypes we predict? And now in boldface are the ones that match up with the ones that Marianne observed, not only in C. elegans, but also in C. briggsii and C. remini. So this comparison isn't across all 11, but 3 of those 11 that she studied. So we have a far greater hit rate if we look at all of these other, the 3 other species of related to C. elegans. But there are still some gray ones that are not overlapping with C. elegans, C. briggsii, and C. remini. They may be in the other 8, but they could be entirely misses by the model. So it's certainly not intended to suggest it's a perfect model, but we do better if we compare across different species. And so this leads us, I'll skip this slide, this leads us to confirming that C. briggsii, C. elegans, and C. remini may indeed share a same network that's quantitatively diversified, and that's sufficient to prove that that would give rise to differences when you perturb them away from wild type. Okay. And in a model, you can do something you cannot do in an experiment, which is you can now ask, well, what is different about C. briggsii and C. elegans? Because you presumably narrowed down what parameter points allowed C. briggsii to have access to orange uniquely, whereas C. elegans has access to pink. Okay? And when you do that comparison, you get these kinds of results, where you can compare, I'm just going to focus on this right panel, C. elegans to C. briggsii, and identify among the 8 parameters in the model which one has changed, or which ones have changed, and in what way. Have they gone up, empty bar, or have they gone down, black bar? To explain qualitative phenotypic data that's identified in C. elegans. So here, it's very clear that there's one parameter that's different, and it has to be higher in C. elegans than in C. briggsii. It has to be higher in C. briggsii than in C. elegans. In the comparison with C. remini, there are 2 parameters that stand out starkly as having to have changed in a particular direction. Okay. And I leave the diagram below because it reminds me to make the point that these Greek variables are not esoteric variables. They map back onto molecular mechanism. They tell you which arm of these molecular mechanisms must have gone up or lower in strength during evolution. So I'll close with this, with 2 slides that summarize what we think we are observing in these systems. One is that in the context of C. elegans, you certainly have networks that define phenotypes. Okay? And one can think about evolution occurring as the topology of this network changes. It gives rise to access to new phenotypes. You add a feedback, you eliminate a pathway, and so forth. But what we've been exploring is what happens on the quantitative axis. Fix the network, vary the parameter intensities, and we would argue that there is a significant amount of diversity that can occur along that axis, and it's diversity that seems to exist in record, for at least for the C. elegans and related species. And what you can do with a model such as this is to back out what those changes must have been. And we think that this is intimately related to what I originally started talking about, which was the epithelial cell system. Cancer is an accelerated evolution of a biological system. And if quantitative variations are important in this situation, they could very well be important here, as we've shown in the context of stiffening the matrix. So understanding what those quantitative interactions are and how to think about phenomena like contact inhibition with a quantitative framework could be very useful in understanding cancer progression. And ultimately, these quantitative interactions and rules play out over multiple scales, at the molecular and physical level in terms of signals, but affecting cellular behavior and then multicellular structures. So whenever you have these types of situations of multi-scale, you of course have to be thinking integratively of how to weave these scales together in an informative way. And clearly computational modeling is a core of this effort, and we envision that similar models could ultimately be developed for epithelial cells, and we're striving to understand experimentally first what the building blocks would be for such models. And en route, as we understand contact inhibition, motility, and other processes, we think we're contributing to this idea of tissue engineering and the design principles needed to enable tissue engineering in a guided and rational fashion. So with that, I will close and thank and acknowledge the people who've done the work. Jin-Hong Kim has been the driver of the epithelial proliferation work. Kei Kushiro is looking at cells moving on micropatterns. Melissa is doing the aggregation work that I described. Paul Miner has been following on Claudiu Ghiromescu's work. Claudiu did all the worm modeling work that I described in collaboration with Paul Sternberg in biology. And we're starting a collaboration with a group in mechanical engineering to measure some of the forces that these epithelial cells exert on the soft substrate underneath. And I'd like to, of course, thank funding from various sources. And in particular, because I'm here at USC, I would like to draw attention to this one, because I think this is going to now push us in this realm of multi-scale computational modeling, now extending not only from molecular cellular levels, but to organismal lymphoma models that are being examined in mice. And we're going to be involved in that computational effort to develop what we're calling a virtual cancer model for lymphoma. So I'm happy to entertain any questions you might have. And again, I'd like to thank Michael and the department for an opportunity to visit. Thanks.
The model you presented, unless I misread it, was
relatively simple. It was one-dimensional, it felt, actually, and the way you analyze the predictions are by completing simulations to the end. I understand that's how you probably do it. But when you get to a model of the kind you are describing first, epithelial cells, other kinds of tissues that are a lot more complex, you're going to have 2- or 3-dimensional interaction between many more signals than the ones you actually put, probably leading to a lot more possible phenotypic types. And I just wonder whether there is computational foundations to actually understand this without just doing the experiment and seeing what you get, because you're going to get more phenotypes, is my feeling. I might be wrong there.
Yeah. So I think there are multiple layers to that question, so I'll try to hit on some of them. So a clarification first on the C. elegans model itself. Indeed, it's one-dimensional spatially. That's motivated by the physiology of the system. We thought about, and you're right that we didn't model transport in that system, and that's because the transport step, the time scale of it, versus the time scale of the decision-making process is much faster. So we assume that the gradient that we're talking about reaches a steady state that then encodes the decision-making process. So the gradient is encoded as a parameter that defines the decay from the center. The model does look at the steady state of the state variables, and then uses that to predict what the fates of the system would be. So we don't quite run it to the end point. That is, we don't bother with running the dynamics if we're going to be interested in steady state, but we are looking at a steady state endpoint. So those conditions seem to work, but doesn't necessarily mean it's the only way to conduct even the C. elegans simulation, because one can think about the fact that perhaps dynamics is important. Perhaps the system is choosing its fate early on. The endpoint is not when it chooses the fate. So we've run simulations that follow the situation up to what is a physiologically relevant time scale for this system, which is a 6-hour period. The reason we choose 6 hours is we know that if you eliminate critical signals before that time, the fate choices will be wrecked. And so we know that choice making is still going on up to 6 hours in the system. And when we do that, we get very similar results. So looking at transient behavior as well. But in terms of extension to more complex systems and what might be needed in terms of computation, that is, let me answer it in 2 ways. The first is sort of a semi-cop-out way, which is to say, well, we have to get there to sort of know what the hurdle of complexity is going to be involved. And we don't think the hurdle is going to be to, and this gets at my second answer, which is we don't think it's going to involve simulating these massive networks that you see pictures of, the complexity, the classical complexity picture, because, and this is just our philosophy and how we approach the system, which is we tend to reduce the system to principal players, growth factor, EGF, MAP kinase, even the C. elegans model. It could have been far more complex than the one we used to make the predictions because we know a lot more. We know a lot more arrows and molecules that are involved in this crosstalk than was put in the model. We use the coarsest description of the system. So I think coarse graining the description of these networks is going to help give access to what would otherwise could be a computationally complex scenario. The coarse graining is, in my reference to taking complex sketches of the networks that you typically see and saying we can collapse certain pathways into a singular effect. And that's the type we did with the C. elegans. If we didn't do that, I don't think we could have explored parameter space the way we did, because we were able to do it because it's an 8-dimensional space. If I represented all the details of the pathways in that model, it would not have been a feasible project to undertake. Yes?
Once you translate this C. elegans model to a more complex system in which not only are you dealing with genetic factors, but you're also dealing with epigenetic cues, as well as extracellular cues in the context of tissue engineering and cancer.
I don't know that the C. elegans model, as configured, is directly plug and play into another biological system, even one that's influenced by EGF and Notch delta, because, like you said, the EGF and Notch delta could be interacting in a different way, in a different set of pathways, or there could be other factors that we haven't accounted for in the C. elegans system that's relevant in the human cancer system, for example. So we don't. I wouldn't want to suggest that we believe that the model performed well and therefore can be applied as is to any other situation, because the approach might be applicable, but the details of the model will probably have to be revisited because there could be other pathways that are important. But in the context of the epithelial and other system, which I would suggest is more complex than the C. elegans, this is why we're trying to do the experiment first, because we need to know what the relevant environmental signals are. So compliance, EGF, coherence, we know generally that they're important, but how they quantitatively impose and compete against each other is not so clear. So we try to emphasize the experiment first, elucidate sort of like a state diagram framework that I described, which could then help us build a model that's relevant for that system. So we are in the process right now of developing a lattice model for the epithelial cell system based on what we've learned about the relevant molecules, EGF, coherent, actually cell shape is involved in that model, and compliance of the matrix. So did I answer your question?
I'm not sure what I'm asking, so I'll just try.
Can I use that for my answer as well?
Yeah, you can. I'm wondering what this is going to look like in 20 years. I mean, what sort of
Yeah. No.
architecture, they handle
Uh-huh.
maybe numbers, or I don't know what they do.
Yeah.
What's your idea?
Yeah. So, looking at 20 years and then asking where this would stand. Let me answer it in two ways. One, I think, is that as we study, for example, these systems, other systems, we might begin to learn that what I studied here as an E-cadherin EGF interaction, crosstalk, and a threshold property for proliferation might emulate as a crosstalk between N-cadherin and NGF for neuronal differentiation. But yet, the overall architecture of saying there are competing cues with a state diagram of this type and a threshold concept may hold. And that allows you to say, well, there's that allows you to abstract away the details of the molecules and the function and say that there may be competing molecular cues that work through a threshold property, for example. And what those specific players are and what behavior is being thresholded and so forth may vary from system to system. And to understand that and to know if that's true, we may need to prove by example, maybe for 2, 3, or 4 different systems. But those examples may move us to a more general paradigm for how we can think about functions of a cell. And with those more abstract rules, you might then be able to think of models where you just have Q1 and Q2 that are competing cues and so forth, and you can run a simulation where you're not even you might be thinking about evolution of the system not so much on a spatial axis per se, but it may even be an abstract axis that these cues compete for a function that you care about. But ultimately, when it comes down to application, we're going to be interested in specific systems and specific molecules and specific structures. And so I think both are going to be needed. Advances in those 3 or 4 examples are going to maybe distill into a common principle. But we're also still going to have to know that this is for epithelial cells, and for neuronal differentiation, you need to think about other molecules, because that's the way you can get into a practical application.
We're going to understand the dynamics of an organism,
Yeah.
particularly a multicellular, multisystem organism like humans. The principles are going to really stay.
Yeah, yeah, I think so. I think the principle might be the.
Very different from what it looks like now. Not that,
Yeah.
I'm
Yeah.
trying to think of what they
Yeah.
look like. Maybe it's enough.
No, I think that's right. If this idea of a state of a thresholding property like this that I just described carries out into 2 or 3 or 4 other systems for different functions, you may at that time point, at that snapshot in the 20 years, call that a distilling idea, a possible principle. I hesitate to put such big words on stuff. And then, maybe that's it, and there are other principles like that. But I think time will tell if this qualifies as something of that type. An example might be morphogens. Morphogens in development, that spatially graded growth factors giving rise to decision points based on a concentration, local concentration. That was speculated as a paradigm a long time ago. Experimental examples came after that, and the proof has shown to be true, but it's not that morphogens work by themselves. There are a host of other mechanisms that help to entrench the pattern, but it's certainly a pretty good paradigm, a pretty good principle for achieving patterning that seems to recur over and over in a variety of contexts.
Thank you very much.
Thanks.